Hi, I'm Joe Bauer from the University of Pennsylvania, and I'm going to tell you about some of our work and of others in the field of NAD metabolism. First, a couple of disclosures. These are companies that have either sponsored some of the work in the lab or who I have consulted for. And to get started, this is the general interest of my lab, which is what causes aging and whether there's anything we can do to slow it down. So to take someone with this sort of less successful aging trajectory on top and redirect them to a more successful aging trajectory on the bottom. And the best way that we know how to do that right now, at least experimentally in laboratory organisms, is caloric restriction. So this is a survival curve from a typical calorie restriction experiment where you just simply give the animals less calories. It's female mice in this case. And the inside curve here is the control group fed under normal conditions that you would raise mice under. If you mildly restrict their calorie intake by about 10%, you get the survival curve shown here in the middle. Where you get a small but significant lifespan extension. And then if you very severely restrict their calorie intake by about 50% in this particular experiment, usually about 40%, you get this pretty dramatic shift in the survival curve here, where even the luckiest animals in the control groups are dead before the majority of animals in the calorie restricted group. So this can have a really profound effect on lifespan, and it's really accepted at this point to say that calorie restriction really does affect something fundamental about the aging process, not just a few specific causes of death. And so we've been generally interested in how this might work and in many different topics related to that. And that was really the driving force for my lab to start thinking about NAD. Because this is one of the things that responds to calorie restriction. Of course, there's many other routes to getting to NAD, and many of the other labs that work on NAD never thought about it this particular way. But for us, it was these observations in papers like the ones shown here. So this is skeletal muscle NAD content. If mice of different ages, so 6 months on the left here, or 22 or 30 months, which are much older mice, near the end of their lifespan, and you can see the skeletal muscle NAD content is falling pretty dramatically. And on the right-hand side, you can see that if you calorie restrict and then look at aged animals that were calorie restricted versus not, you can see that that's restoring the muscle NAD content. And we see the same thing with exercise. So there's generally a correlation between the NAD content of tissue and these interventions that promote health and longevity. We've done some work in my own lab in collaboration with Josh Rabinowitz's now to look at NAD levels across a variety of tissues. And so this is just data in our own hand showing that in many tissues, NAD levels do seem to fall. This is 25-month-old mice compared to 4-month-old mice. And you can see in liver, kidney, and skeletal muscle, the quadriceps here, we see decreases in NAD. It's not universal. In our hands, it's not so clear that it falls very much in the pancreas or spleen or stomach. But we do definitely see a decline in NAD levels in many tissues with age in mice, and others have reported the same thing in humans. There's sort of a growing body of literature on this now, but clearly it's true in the skin, which was the first tissue reported for humans. We were involved in this study with colleagues here at Penn and in Ravindra Reddy's lab, who were able to show that there's a correlation between NAD concentration in the brain and age by magnetic resonance spectroscopy, and in liver biopsies from humans, the same thing has been reported, where the people over 60 have much lower NAD levels in the liver than people under 45. So that's why we're interested in NAD, and I think before we continue to show any more data on the subject, we'll stop and remind everyone exactly what we're talking about, which possibly you remember from biochemistry textbooks. But it's this molecule shown here in the center, nicotinamide adenine dinucleotide, that plays critical roles in redox metabolism. And so that's shown on the right. It's involved in things like glycolysis, beta oxidation, the tricarboxylic acid cycle, where NAD accepts a hydride ion, so electrons and an H+, to get reduced to NADH, shown on the right. And then that gets reoxidized by processes like donation to complex 1 of the electron transport chain to fuel mitochondria, by the lactate dehydrogenase reaction, or many other dehydrogenases that can use these electrons in different biochemical transformations. And so that cycle is really fundamental to life. We really have no sustainable route to generate ATP or to synthesize many macromolecules without having this ability to shift reducing power back and forth through NAD. Now, on the left side, I'm showing consumers of NAD, which are more recently discovered. They're important in their own right, things like PARPs, CD38, sirtuins, which have critical signaling functions and regulate different aspects of cellular physiology. But importantly, they actually consume the NAD backbone and release nicotinamide. And so this necessitates a salvage pathway to resynthesize NAD from nicotinamide. And it creates the possibility that these things can get out of whack. And so the size of the NAD pool can change if the consumer activity gets upregulated inappropriately, or for some reason salvage synthesis is blocked. And that affects both sides of the slide. Because once the NAD pool falls, then it's no longer available for these redox reactions, even though the redox reactions themselves don't consume the NAD. They allow you to regenerate it. And so even from what I've already said, I think it's clear that NAD is completely essential for life. If you don't have it, that's going to be lethal. We actually have human data to prove that, because it turns out that a deficiency for NAD precursors is the cause of the disease pellagra, which was endemic in the southern US in the early 1900s, and leads to what was known as the 4Ds: dermatitis, diarrhea, dementia, and death. So if not treated, this disease really can go on to become fatal, even when you obviously still do have some NAD levels present in the body. So this discovery that pellagra is caused by an NAD deficiency is integrally related with the discovery of NAD in the first place. And so I'll give you a little bit of history on that, starting with the discovery of coferment, which was by Harden and Young in 1906. They were doing experiments with boiled yeast extracts where they discovered that there was a substance present in the yeast, even after boiling, that could accelerate the fermentation of alcohol. They named it coferment, not really knowing anything else about it at the time, other than it was something that could survive boiling. Between 1914 and 1929, Joseph Goldberger was active in trying to establish that pellagra was a nutritional disorder. It had been debated whether it was an infectious disease, and that the cause hadn't been understood. And he was actually able to demonstrate that if you improved the quality of the diet of people suffering from pellagra, particularly adding meat and milk, you could cure the disorder. In the 1920s, Hans von Euler-Schelpen discovered that coferment is actually a nucleotide. So this substance that can accelerate the fermentation of alcohol was something that had a nucleotide structure, still not knowing what it was. Warburg and Christian went on to determine that it actually contains a nicotinamide moiety, which is what actually supports the redox reactions, since nicotinamide, when attached to a backbone, is capable of accepting the hydride ion. And finally, Conrad Elvegem and colleagues put it all together at the end of the 1930s, and were able to show that nicotinamide and nicotinic acid can prevent pellagra and contribute to the synthesis of this redox active nucleotide, which turns out to be NAD, obviously. And so that was a real tour de force, and established nicotinamide and nicotinic acid as vitamin B3, something essential that we need to prevent the onset of pellagra. And obviously facilitated curing this disease effectively in Western countries that could afford the vitamin supplements. So since then, the question that's been left to be answered is, do the NAD levels still matter once you've avoided pellagra? After the 1940s, pretty much you were considered to have sufficient levels of NAD if you weren't suffering from pellagra. And so it's really only recently that we've revisited this question and asked whether there might be some benefit to boosting NAD levels in people that are apparently healthy. And there's a few different reasons to think that might be the case. One of them is that NAD levels are circadian. And so at least in the liver and in several other tissues, our body has seen fit to put a lot of effort into regulating the levels of NAD biosynthetic enzymes here on the top. This is nicotinamide phosphoribosyltransferase, and the black trace shows the nice circadian rhythm in a wild type liver. The white dots show a clock mutant. So if you break up the circadian clock, you also lose rhythmic expression of NAD biosynthetic enzymes. And on the bottom, you can see the result that the NAD levels are actually going up and down in the liver over a circadian time course. In addition, there's a growing body of literature showing that in humans, NAD level doesn't just correlate with age, but also with functional status. So really suggesting that there might be a real benefit to having a little bit higher level of NAD even within the normal range. And so the large metabolomics graph over here on the left is just highlighting the fact that if you rank metabolites based on either the age or the functional status of individuals, NAD really shows up as something that gets downregulated in either case, either by age or functional status, and stands out among all the other metabolites that were measured in this particular study. The inset up here on the right makes another point, which is that functional status really plays a critical role here. So if you look in the older adults and then the three groups on the right, you can see on average there's lower NAD levels than in the younger adults as expected. But if you look at exercise trained older adults here, the second group, their NAD levels in the muscle actually aren't even statistically significantly different from those of young adults. And so this is something that you can potentially modulate through exercise and healthier habits. And it's something that really reflects the functional status much more than the age per se. And so based on studies like that, we and many others have been interested in asking whether life's really better with more NAD. If we can boost NAD levels, will you be healthier and better off? And the original ways to do that are shown here. These are the structures of nicotinamide and nicotinic acid, which collectively receive the designation vitamin B3. You'll also hear nicotinic acid referred to as niacin, and that name was provided just to avoid confusion with nicotine, shown here on the right. These are not the same thing and not really related to each other, just has a coincidentally similar sounding name. And on the bottom, I'm showing tryptophan also because this is a starting point for a de novo NAD biosynthesis pathway that was worked out in the 1940s after it was realized that tryptophan could also be a substitute for vitamin B3 and give you an alternative way to generate NAD levels. What I'm really going to focus on for most of my time is these newer NAD precursors. First of all, nicotinamide riboside, which was discovered only in 2004 to have a kinase that allows it to directly contribute to the NAD synthesis pathway by being phosphorylated to generate nicotinamide mononucleotide. Previously, it was known that this substance existed, but it was thought that it would have to break down nicotinamide and then go through the normal NAD salvage pathway in order to contribute. But the discovery of NRK kinases showed that there really is an alternative way to get here to nicotinamide mononucleotide. And at the same time, there's been a growing interest in just directly supplementing nicotinamide mononucleotide itself. And so I'll talk a little more about both of those as we go on. So this is the NAD salvage pathway that I've mentioned a couple of times. So here's NAD, different consumers release nicotinamide. Nicotinamide phosphorybosyltransferase is the rate limiting step that generates nicotinamide mononucleotide. And then enzymes called NMNATs, NMN adenylyl transferases, they catalyze the last step where they add an ADP ribose phosphate onto the nicotinamide mononucleotide and regenerate NAD. And you can have input to this NAD pool also from tryptophan through the de novo synthesis pathway, from niacin through the Preece-Handler pathway. And then as I highlighted on the previous slide, it's now recognized that with nicotinamide riboside, you can come in from the side here through NRKs and generate the NMN pool through an alternative way. And importantly, this bypasses nicotinamide phosphorybosyltransferase, which requires more energy and is rate limiting. So you can potentially boost the NAD pool higher by supplementing nicotinamide riboside. So a couple of groups went on to do that immediately. And this is one of the first papers showing the effects of nicotinamide riboside in mice. And what they showed was that basically a big boost in mitochondrial biogenesis occurred. And so on the left side here, I'm showing the running distance for animals supplemented with nicotinamide riboside. So this is without training. If you just put them on a treadmill, they're able to run farther if they've been receiving nicotinamide riboside. And that was at least partly attributed to the fact that mitochondrial biogenesis is upregulated in muscle. And so these are a variety of transcripts related to mitochondrial biogenesis, all being upregulated by the nicotinamide riboside. So papers like that spurred a lot more research over the past decade. And there's actually a lot more benefits of NAD precursors that have been reported than I can really go through in this lecture feasibly, but I thought I'd summarize a couple of the themes that have been driving human clinical trials. And so one of them shown here on the left is improvements in glucose homeostasis and insulin sensitivity. So this is a glucose tolerance test where you give a bolus of glucose and watch the blood sugar go up, and then how fast the animal is able to control it and bring the blood sugar back down. And you can see here in black, these are aged animals that were not able to control their blood sugar very well. It stays pretty high over the course of 2 hours in black, but if they've been given a 1-week course of nicotinamide mononucleotide to boost NAD, you can see their glucose tolerance and clearance is much better, and they get back down almost to normal by the end of 2 hours. The middle graph shows an example of heart failure. So there've been a variety of different models used to show this now for NAD precursors. This particular one is serum response factor knockout. That is a model of dilated cardiomyopathy. And what the graph is showing is fractional shortening. So this is similar to ejection fraction, just a simpler way to measure ejection fraction when you're doing echocardiography in mice. And if you look at the white bars here showing just the regular diet-fed mice, they've lost about half of their fractional shortening, so about half of the contractility of the heart by the time that this experiment was done. And if the animals were receiving supplementary nicotinamide riboside, you can see a little more than half of that loss is prevented. So you're quite a bit closer to the control level of heart contractility with NAD precursors on board. And on the right, I'm showing an example of a cognitive model. So there have been a variety of different Alzheimer's disease models now shown to be improved by NAD precursors, but this was one of the first papers to show that. And they're doing a novel object exploration paradigm. And so the control animals here not receiving any supplement have Alzheimer's disease at this point and are impaired to the point where they can't even beat chance performance here. So not even 50% of the time are picking the novel object to explore. But if they've been given nicotinamide riboside to boost NAD levels, you can see here in the black bar that actually gets them back to being able to discriminate which of the objects is new again. So again, a promising improvement in cognition that is leading people to really look at doing human clinical trials to look at cognitive function. There's also been some encouraging results in terms of cachexia or muscle wasting. So this is a cancer model where animals have been implanted with a tumor that causes them to experience cachexia or wasting. And you can see the body weight loss here in the control group dropping right off by 21 days after administration of the tumor. And at 2 different doses of nicotinamide riboside, you can see that weight loss is pretty well attenuated. And we see the same thing on the right. If we look at the leg muscle mass, it's dropping off in the control group, but nicotinamide riboside really helps maintain muscle mass, which we hope is encouraging for aging humans with sarcopenia. There have been a couple studies in actual aged mice, to not just disease models, but really looking at naturally aging mice to see if there's any improvements from boosting NAD levels. And they've been generally encouraging. So we look at things like bone mineral density here that are going up with 2 different doses of nicotinamide mononucleotide. Triglyceride levels in the liver, at least at the highest dose, are dropping off. And so there's evidence for improvements in metabolic function in the aged animals with NMN. If we look at nicotinamide riboside in a naturally aging group and then look at the muscle stem cell pool here on the top, you can see it actually has dropped off, comparing the gray bar to the white bar in the aged animals. And in either case, with nicotinamide riboside supplementation to boost NAD, you can improve the number of muscle stem cells and the muscle regenerative capacity in the aged animals. On the bottom, I show from the same study, it was shown that there was a statistically significant extension of overall lifespan when nicotinamide riboside was started at a high dose late in life. You can see from these survival curves that the effect size was not that large, but it was statistically significant in this first study. But having seen that, I want to also point out a later study, which was done at 3 different centers through the NIA's intervention testing program, and included both males and females. And so, albeit at a lower dose of nicotinamide riboside, this was a much more comprehensive study starting earlier in life. And here there was no benefit at all seen on overall survival. And so I think this is an important caveat to keep in mind, that there's a lot more evidence for nicotinamide riboside or for other NAD precursors helping with disease processes than for extending overall healthy lifespan. In the same vein, as we've started to move into human clinical trials, it's important to recognize that there have been a lot of negative studies for some of the same indications that we've seen in mice. So this is one of the early ones shown here on the left, looking at insulin sensitivity, where lots of different rodent models have shown improved insulin sensitivity, but this is glucose infusion rate. In humans, either placebo-treated or NR-treated before and after the intervention. And you can see they're all completely overlapping. There's really no improvement in insulin sensitivity from this NR trial. The only thing that came out of this one, in fact, that looked a little bit promising was a non-significant decrease in liver fat that was seen in the humans. And so that was considered something interesting to follow up with longer studies in the future. But really the goal here was to show improvements in insulin sensitivity and glucose homeostasis that were not seen. Some of the other trials have actually missed their primary endpoints, but then made other interesting observations. So this is one of the examples of that. Here, they were looking for improvements in muscle strength and mitochondrial function. And on the top, you can see that there really weren't any. These are, you can see the labels over here. The blue is the NR treated, and purple is placebo, and black is a baseline evaluation. They're all overlapping in terms of muscle oxygen consumption, so mitochondrial function. And the same thing for hand grip strength, shown here on the right. There's really no change at all. But during this study, they also noted that there was a decrease in inflammatory cytokines. So you can see IL-6 here and IL-5 and IL-2 are all lower in the blue bars treated with nicotinamide riboside, suggesting that it might really have some anti-inflammatory activity in humans. And another couple of comprehensive studies that looked at a variety of phenotypes. Most were coming out negative, but there were reports about body composition improving. And so one example of this is shown here. This is fat mass and fat-free mass. So you can see fat mass is actually decreasing significantly to a small degree, but significantly, and it's being replaced by lean mass, such that body weight's not changing. But this is definitely seen as a favorable change in body composition to convert fat mass into lean mass, which was seen in this study. And I believe also in the next one that I'm going to show you, which was, I think, the most promising trial to date of NAD boosting in humans. And in this case, it wasn't just to look at general health, but to actually treat patients with mitochondrial myopathy. They also chose niacin, so nicotinic acid, rather than riboside or mononucleotide for this particular study, and did a pretty high dose and went as far as 10 months. And so this is one of the most comprehensive studies of NAD boosting that's been done. And importantly, they did hit their primary outcome, which was functional tests. So the 6-minute walk test is shown here as an example on the left. As expected, the controls were pretty good at doing these sorts of basic functional assays and not significantly affected by the supplementation. But the patients who were impaired in the 6-minute walk test showed a statistically significant improvement over the course of the experiment, indicating that niacin really may be therapeutic in this disease, and certainly spurring larger studies. At the same time, in both the patients and the controls, you can see that there's a favorable shift in body composition, as was seen in the previous study. There's a loss of fat mass and a corresponding gain in muscle mass, suggesting that there really may be a benefit to NAD boosting in terms of converting fat to muscle. So to summarize the human studies to date, I think there is some support for anti-inflammatory effects and improvements in body composition, but really inconsistent or negative results mostly for liver fat, blood pressure, insulin sensitivity, muscle function, and mitochondrial function in normal aging. So many of the same indications where the rodents did suggest there might be a benefit. As I said, generally no improvement in mitochondrial function. And there are, I think, pretty promising results in this mitochondrial myopathy trial, again, suggesting that maybe treating diseases is going to be more impressive with NAD precursors than just trying to improve general health. So there are a number of outstanding questions that I want to highlight for the field that we're really having to deal with now, since so much of the work has been done just by giving these supplements, boosting NAD in the whole body, and then just looking at outcomes. We haven't learned all that much in most of these cases about which of the downstream pathways matter. There's hundreds of redox reactions that NAD is involved in, and at least tens of these different enzymes that break down the chemical backbone and consume it as a co-substrate. And in very few cases do we have real evidence as to which one is important for the effects that we're seeing. I'll show you one just cautionary note on this subject, which is that when you see this pattern, there are a number of papers out there concluding from this pattern that the given enzyme is mediating NAD's effects. So here we're looking at wild type animals, with regenerating livers. And we had shown previously that NR can boost liver regeneration. You can see the liver looks a little brighter here, it has a healthier color, it's a little farther along. And if we do the weights, you can see that NR allows faster regeneration. So we were curious whether this involves a specific NAD consuming enzyme, SIRT3. So we got the null mice and we did liver regeneration assays on those mice. And you can see here that they regenerate poorly. So this liver is filling up with fat instead of making new liver tissue when it's trying to regenerate, and generally looks terrible. But if we actually give them NR, it almost completely rescues that phenotype. So it's not that NR is actually dependent on SIRT3, they're actually having independent effects. And we've seen this also for SIRT1 in these liver regeneration assays. That's also important for regeneration, but independently from the effect of NAD boosting. In fact, here, if you look at the mitotic index, so how many hepatocytes are replicating, you can see that the SIRT3 null mice treated with the nicotinamide riboside actually have an even bigger benefit than the wild type animals do. So I think it's very important to do the full experiment and really test some of these pathways that people hypothesize are mediating the effects of NAD precursors, not just assume from what looked like similar effects of knocking out a specific gene that is necessarily the pathway that's relevant for the NAD boosting that you're seeing. Another critical question is which tissues or cells or even organelles are really mediating the effects of the NAD supplements. And as I said, in many cases, we're putting these in the whole body. And so we don't really know if it's a direct or an indirect effect that's leading to whatever we're measuring. And so one way that we've tried to get at this is to target this enzyme, NAMPT, in specific tissues. So we have a flox mouse, and we can knock it out in a tissue of interest and lower NAD only in that tissue and ask what the consequences of that are. One of the first experiments we did in this area was to knock it out in skeletal muscle. And so this top band is NAMPT. You can see it's completely gone from different skeletal muscles after we've tried to knock it out. And that actually turned out to have a very dramatic effect on NAD levels. We thought some of the other pathways might compensate a little bit, but these animals have NAD levels that are somewhere around 15% or 10% of normal. And it turns out they don't die right away. In fact, they look kind of normal for a surprisingly long time, despite having very low NAD levels. So what you can see here is one of these animals that has very low NAD in their skeletal muscle at about 7 months of age. And by this age, I hope you can tell which is which. The animal with low NAD is kyphotic. It has this arched appearance and it doesn't move around very much. And as expected, they do much more poorly in a variety of measures compared to this wild type littermate that is running around acting normal. And so if we look at those animals, I won't dwell on the phenotype because this was published a while ago, but what I want to highlight is the point where they really start to have dysfunction is the point where mitochondrial respiration becomes impaired. And so that's shown here on the left. These are data from a Clark electrode making different additions. So you can really focus on this set of bars here, which is state 3 respiration. And you can see that in the knockout animals in red, respiration is impaired. And if we've given them nicotinamide riboside, it actually comes back to normal. So their mitochondria function is completely restored, and those animals regain their running ability and appear really functionally normal as well. At the same time, if you look over here on the right, you can see these control animals at the same age where they're having dysfunction, or the knockout animals here on the right at the same age when they're having dysfunction compared to these control animals are still able to produce lactate. So when we run them, the blood lactate actually goes through the roof, meaning glycolysis is still intact. So that's an NAD dependent process, but it's still working. And it's really the mitochondrial dysfunction that seems to be what correlates with their loss of functional capacity. And so partly because of this result, we got really interested in how NAD gets into the mitochondria in the first place. And that led us and a couple of other groups to come to the conclusion that it's through this channel SLC25A51, or so transporter. So we had investigated a variety of different candidates, and this is the one that came up with the phenotype we've been looking for, which is if you take whole cells and look at NAD levels here and a wild type or a knockout for this transporter, there's no effect. NAD levels are totally normal, but if we isolate mitochondria from those cells and then measure NAD, they're drastically lower in the knockout. And we and others have done a variety of experiments to really establish that this seems to be the protein that's involved in carrying NAD into mitochondria. Where we know it's essential for function, and we can show that very dramatically using these knockout cells. So this is oxygen consumption rate. This is a seahorse assay, but OCR is basically just the amount of oxygen they're consuming as they get put through a variety of mitochondrial poisons. So this is the basal oxygen consumption in wild type. If we give oligomycin, this is uncoupled respiration. If we give FCCP, this is, the oligomycin is leak rate, and FCCP is the uncoupled respiration, and we give rotenone and antimycin A just to poison the mitochondria and see that oxygen consumption go away. And it doesn't really matter what that pattern is to compare to the knockouts, because the knockouts essentially just look dead. They're completely flatlining on the oxygen consumption because without NAD, the mitochondria really can't consume oxygen. And so we were able to actually put back the transporter and rescue this. These cells are not dead, despite appearing that way based on oxygen consumption. They actually will go on and proliferate, just deriving all their energy from glycolysis, albeit very slowly. But you can really see that this transporter getting NAD into mitochondria is really essential for this mitochondria to work. So we are in the process of characterizing what this really means in vivo, what happens in animals lacking this transporter in different tissues, and starting to comb through some human data. One of the really obvious pieces of low-hanging fruit is that there was already data in the Human Protein Atlas showing that this gene is prognostic for different cancers. So if you have liver cancer or thyroid cancer, it's a bad thing to have high levels of this transporter, which would be supporting mitochondrial function in the tumor cells. Maybe that makes sense. In renal cancer, it's the opposite. In renal cancer, it's favorable to have a high level of this gene. And that's interesting because renal cancers are sort of famously glycolytic. So they don't depend on mitochondrial metabolism normally. They shift their metabolism to glycolysis. And when you force all the energy into the mitochondria, that actually seems to be a problem for the renal cancers. So that'll be something that's interesting to explore going forward. And so the final outstanding question that I'll spend some time talking about is what is actually happening to NAD turnover flux here at the bottom. So we know steady state is decreasing with age, but there are different ways that can happen. And just measuring the steady state doesn't really tell you whether NAD is being turned over faster or slower, or so are NAD-dependent enzymes actually more active or less active. So to try to get at this, we employed a system that was pioneered by a couple of different people, particularly Ling Lu and Josh Rabinowitz's lab, and then Melanie McReynolds when she was in Josh's lab, and Karthi Chalapa in my lab, used this system to measure NAD turnover in a variety of systems. So what this uses is this tetradeutyrated nicotinamide. So there's deuterium at 4 different positions. And when you convert this into NAD, this position at the top here is the redox active position. And so that starts exchanging and you lose that deuterium pretty quickly. And so you get mass plus 3 instead of mass plus 4. And that's shown over here looking at the NAD fraction in cells over time. Mass plus 0 is the original NAD the cells had. That goes away if you're incubating them with this labeled nicotinamide, and you accumulate mainly mass plus 3. You also can slowly lose a little bit of this deuterium as well. And that's accounting for this mass plus 2, which is a minority component that we don't usually pay too much attention to over shorter experiments. And so when we use this system, we can learn a couple of things pretty quickly in cell culture. In this case, we just inhibited NAD synthesis and watched the decay of the unlabeled NAD. So the label here is really just showing you that we're not making any new NAD from the labeled nicotinamide that's present now. And what you can see from the decay is it follows approximately first order kinetics, meaning that the amount of NAD present is determining how fast it gets used up. So the enzymes that are consuming it are sensitive to the concentration, which means that if you boost NAD levels correspondingly, you would expect to increase the activity of some of these enzymes. On the right, we can show that in these cultured cells, which were a breast cancer-derived cell line called T47D, A lot of the consumer activity seems to be dependent on sirtuins and PARPs, which are some of the well-known consumers, because we can inhibit either one of those. And this F out is the turnover rate, essentially, of the NAD pool. And you can see it decreases pretty dramatically with either PARP or sirtuin inhibition, and about 60% of a decrease in that turnover rate if we inhibit both. I should mention these cells don't express CD38, which is the other major consumer people think about a lot. So we really don't know what's accounting for the rest of the turnover in these cells, or how relevant this conclusion really is to in vivo tissues. And I say that partly because if we look in vivo, one of the first lessons we got is that it makes a huge difference whether you're in vivo or not for some of the tissues. So cell lines derived from intestine here in gray, or cell lines derived from anything really, have a turnover half-life of around 6 to 8 hours for NAD. But if you look in vivo at the intestine, the half-life is about 20 minutes. So much, much faster. And we don't really still know why. The turnover in vivo is so much faster than in the cell lines, but you can see discrepancies in other tissues, and in particular muscle. The half-life speeds up in cultured cells, but it's actually about 14 hours in vivo, so very slow turnover of the muscle NAD pool. So of course we wanted to use this system to ask why NAD is lower in old mice. So we set up essentially the same thing that I've been showing you data from, where we have free-running mice that are getting constantly infused with this tetradeutyrated nicotinamide, now comparing 3-month-olds to 25-month-olds. And we really had two major models in mind, just to set up the way we're thinking about this. The first is that you might have decreased NAD because synthesis actually gets decreased with age, so you're not making as much. In that case, consumption would have to slow down to match the new lower synthesis rate, and this would lead to a decrease in NAD flux. Alternatively, you could have an increase in the activity of NAD consuming enzymes, which would lower the NAD pool because you're consuming more of it. And initially that would result in an increase in turnover. But as the NAD concentration falls, two things can happen. One, synthesis could be upregulated to compensate, and that would maintain the new higher NAD turnover flux. Or second, if synthesis does not cooperate and doesn't change, then what would happen is the new higher NAD consumption will lead to the NAD pool size shrinking to the point where now, because of those first order kinetics, the lower amount of NAD means there's less consumption by the consuming enzymes, and that now balances with synthesis again. And so in this case, flux overall isn't necessarily going to change very much. You might just see what looks like a change in the pool size with very little change in overall turnover flux. And so first of all, the first thing we looked at was nicotinamide MAS+3. So this is nicotinamide that has been through a cycle of getting converted to NAD. So it lost one of the deuteriums and then was released again. So this is sort of an index of whole body NAD turnover. And we're looking in the blood at +3. And the first thing we saw was that in the aged mice, there's at least as much M+3 production. In fact, it looks like statistically significantly more, albeit not too much more, MAS+3 production in the aged mice. So that just tells us right from the get-go that it seems like whole body turnover flux is preserved or maybe even accelerated a little bit with aging, not decreased as we would think if it was impaired synthesis. And I'll blow up one tissue here as an example to really kind of illustrate what we think we're seeing. So this is MAS+0 NAD counts in skeletal muscle. And you can see in the old animals, there's less NAD at steady state, as we had seen before and as we expected. If we look only at the newly synthesized NAD, which is the labeled, the MAS+3, you can see that's actually very similar, and maybe just a little bit more of it appearing in the old, which would suggest that the synthesis rate, if anything, is slightly faster in the old, even though the old actually has less NAD present. So again, arguing for increased turnover flux. Sorry about that. And if we look at it in terms of fractional labeling here on the right, we can see that the total fraction of the NAD that has been newly synthesized in the old, because it's a smaller pool and there's a slightly more MAS+3, the actual fraction that's newly synthesized is much higher. And so the overall conclusion from this is that the smaller pool in the aged animals is turning over more quickly to compensate. So really you're maintaining probably turnover flux, and we can show that on the next slide. So this is the turnover flux expressed in molarity in different tissues in the aged versus young animals. And so in many of the aged tissues, remember the pool size is smaller, but those pools are turning over a little bit faster to really maintain the flux, or in some cases be slightly higher, like in liver here or in spleen. It's a little bit higher in the aged animals. But overall, the main lesson here is that flux is maintained in the aged animals. It's not decreasing, which is what you would expect if it was synthesis that was impaired. It really seems to be that in the aged animals, the model that best fits the data is that consumer activity is increased, and that results in the pool size falling to a point where it balances out consumer activity with synthesis, and you can sort of restore homeostasis. So the last topic I'll talk a little bit about are why such high doses are needed in the rodent studies. Most studies are typically using about 500 milligrams per kilogram of NR, which is about 35 grams a day for a 70 kilogram human if you just extrapolate from body weight. Human studies are usually using about 1 gram, not 35, and that's justified based on arguments about allometric scaling, which is the argument that drugs are typically metabolized much more quickly in rodents, and so you can get away with lower doses as the equivalent for humans. And the fact that there is toxicity that's seen with nicotinamide above about 3 grams a day in humans. And so there's definitely a fear of going to these much higher doses. But human and mouse cells have about the same NAD concentration and the same turnover rate. And so it may well be that you actually have to scale by body weight if you want to get the same effects that we're seeing in the rodents. And so to really try to get a better idea what's going on in vivo, we've employed this tool synthesized by Marie Magote, who's a chemist at the University of South Alabama. And she generated nicotinamide riboside for us that had a label of deuterium on the ribose ring and a C13 on the nicotinamide moiety. So we could track both rings separately. And if this goes straight into NAD, we'll get mass plus 2. If it breaks down, we'll get mass plus 1. And so when we do this, we have delivered NR or NMN either intravenously or orally, and then done a time course looking at first nicotinamide in the blood. So you can see a lot of it breaks down to nicotinamide, but you get a lot more if it was given intravenously than if it was given orally with either molecule. And then if we look in vivo at tissues, you can see here first, if you look at kidney, this green color is the mass plus 2. So this is the molecule that we administered going directly into the NAD pool. And you can see a substantial amount in the kidney with either NR or NMN given intravenously. Not very much, just wisps, if anything, of mass plus 2 when we've given them orally. If we give NR IV, we get the same thing in muscle where you get lots of mass plus 2 contributing. With nicotinamide mononucleotide IV, we actually don't see that. It seems like it contributes much less to muscle when it's given intravenously. And if we give them orally, we really don't see any mass plus 2 appearing at all. And so there's a couple of lessons here. One of them is the delivery route makes a huge difference. So I'm losing control of my slides here. But the delivery route makes a huge difference. Intravenous is clearly much more effective at getting these molecules intact to certain tissues. The second lesson is that there really seems to be something missing when we look at the oral pools. We're not really accounting for some of the administered molecule. It seems like there's less of it everywhere. And one of the things we've recently discovered that might be contributing to that is that the microbiome is playing a huge role in converting nicotinamide that is administered orally to nicotinic acid, or the same thing when you give nicotinamide riboside. You see, so this is the spike in blood levels of nicotinamide here on the left after giving oral NR. And you can see we get a pretty good rise into the 100 micromolar range. And on the right is nicotinic acid. And this is not produced by any mammalian enzyme, but is completely microbiome dependent. And you can see this is actually getting up into the tens of micromolar and 70 micromolar range as well. And that's a high enough concentration to activate G protein coupled receptors that bind nicotinic acid, and to potentially have effects on other enzymes that are independent of NAD synthesis. And certainly it's enough to dramatically fuel the Priest-Handler pathway to make NAD directly from nicotinic acid, which is a different set of enzymes and may have different effects than we've realized from only looking at NR or NAM on cells. So we think this is really important to follow up on and understand whether nicotinic acid production in humans receiving nicotinamide riboside is in this range. So to really get at this in a little more detail, Marie was kind enough to synthesize an even more advanced derivative of nicotinamide riboside for us. So this is a +9 that has all the ribose labeled, so we can track that ring, but then on the amide group labels the oxygen, nitrogen, and carbon. And so we're able to see if this has been deamidated to nicotinic acid by the microbiome and then resynthesized into NAD. We can tell by the mass whether that's happened, even if it's already back in nicotinamide form when we're looking at it. And so Carthy orally dosed mice with this compound. And there's a couple of lessons from this experiment. The first one is if you just ignore the labeling patterns at first and look here at control mice dosed with nicotinamide riboside. This is liver NAD levels. And you can see they go up about 3-fold with nicotinamide riboside. If the mice got antibiotics first, so they don't have a microbiome, then nicotinamide riboside doesn't really work so well. We get a little bit here of mass +9 in purple. So there's a little bit of a rise, but it's not even statistically significant anymore if they've had antibiotics on board. And instead, the majority of this NAD boosting effect of nicotinamide riboside is coming through these mass +3 and mass +1 forms, which are the molecules that show that the nicotinamide riboside broke down, was deamidated by the microbiome, then was taken back up and converted to NAD. And so there's this huge effect that was not really recognized that is being played by the microbiome when you take these supplements orally. And a big part of them is going through a nicotinic acid intermediate rather than staying as the parent molecule or even nicotinamide. So this has some important consequences because a big part of the argument for using these things has been the energetic efficiency. So this is the energetic cost of going through the standard salvage pathway from nicotinamide, cost about 4 ATP. If you give nicotinamide riboside, you can get there with about 2 ATP because of this nicotinamide riboside kinase, which is a more efficient reaction. And when we give molecules intravenously, this is kind of what we see. The majority of it does go through the nicotinamide riboside pathway as anticipated. We see some going through the NAM pathway. But when we give things orally, which is the main way they're given to humans, you can see that there is actually, well, first of all, a lot of it's unaccounted for. There's less going through nicotinamide and less going through the nicotinamide riboside. In fact, very little going through directly through NRKs, through this green pathway. And instead, we're actually realizing that the majority of the delivered bolus is going through nicotinic acid through this alternative Priest-Handler pathway to get to NAD, which is actually energetically less efficient. It takes more ATP to go this way. And may have other consequences, as I alluded to, in terms of activating G protein coupled receptors and things like that. So I think this is also really going to be important to the interpretation of human studies using NAD precursors to really understand how this metabolism is all working. In addition, this got us looking at tracers in mice related to the microbiome. We ended up doing some studies that suggested that even when you don't give this orally, when you give it intravenously, you can end up with a label being converted by the microbiome. And so we did this experiment where we didn't actually administer any exogenous precursor. We're just looking at normal metabolism now. And we've generated mice that were antibiotic treated, so they have no microbiome. That's the ABX. And they're on a diet with or without nicotinic acid present. And we're measuring nicotinamide in the colon lumen. And you can see here, when you wipe out the microbiome with antibiotics and take all the nicotinic acid and nicotinamide out of the diet, you actually end up having an increase in nicotinamide that's present in the colon lumen. And we see the same thing with germ-free mice. So these are mice that were ex-germ-free that were given bacteria again on the left, and then compared to germ-free mice looking at the colon lumen, there's higher levels of nicotinamide in there. And so it seems like the microbiome is actually consuming nicotinamide, and we really at this point had no idea where it was coming from. So we did a series of experiments to show that it actually is coming from the bloodstream, from the host. It's not present in the diet. There's no microbes there to make it. That was really the only explanation that was left. And so we did intravenous administration of this deuterated nicotinamide form, and then looked in the colon lumens, the ileum and so the small and large intestinal lumens of mice that were, again, germ-free is the GF here. So these are germ-free mice that were not administered on the left, that were administered the tracer on the right. And you can see that tons of nicotinamide gets into the colon lumen from the bloodstream. If we've conventionalized the mice, this is decreased quite a bit how much nicotinamide we see there because it's being metabolized by the microbiome. Kind of the same thing in the large intestine. And the reason that we're seeing this nicotinamide disappear in the mice that are conventionalized that have a microbiome is because the microbiome is converting it all to nicotinic acid. And so here you can see, again, nicotinic acids piling up in those mice after intravenous administration of nicotinamide. And that nicotinic acid contains the label. So we know that it's actually coming from the intravenous nicotinamide that we administered into the bloodstream, and it's getting secreted into the colon lumen, then converted to nicotinic acid. And so this is the cycle that I essentially just described. So conventionally, we think that nicotinamide or nicotinic acid come from the diet, and the microbes might get a chance to interconvert them while they're in your gut lumen, and then you would just absorb both and have them in the body as vitamin B3. We do know that you can absorb nicotinamide, but here we show that really the net flux under normal conditions is out. There's more nicotinamide going into the gut lumen than being absorbed from the gut lumen. And in fact, it's generating this cycle where nicotinamide gets secreted into the gut lumen, then converted to nicotinic acid, and then reabsorbed by the host. So there's a sharing of NAD equivalents between the host and microbiome. So finally, we were curious about how important this pathway is to microbial NAD synthesis. It wasn't really clear where microbes get their NAD precursors in the case where you take them out of the diet. And so we decided to test that by doing the same type of experiment. Now, instead of a bolus, we do constant infusion of intravenous deuterated nicotinamide, so we can track this into the gut lumen. And so here you can see steady state levels being reached for NAD labeling in the microbes, which are the main NAD content of the gut lumen. In the small intestine, it's a little bit higher than in the colon or in the stomach, but in all cases, pretty significant. And if we take into account the serum labeling of nicotinamide shown here on the left, which is about 40% on average or so, reaching about 50% by the end of the experiment. If we account for that, we can calculate that about 80% of the NAD present in microbes in the lumen of the small intestine actually originated as nicotinamide in the host circulation. And so this really seems to be the major pathway that's providing NAD precursors for the gut microbiome, at least in the small intestine, and a major pathway in the large intestine as well, where we see a lot of the balance being made up by synthesis from fiber. And so just to go through some conclusions, I think that boosting NAD is an effective strategy to treat many conditions in mice, but studies in humans to date have shown weaker or no effects for some of the same indications, although positive effects in things like mitochondrial myopathy. And so we think it's a very exciting time to be looking at human studies, but also to be thinking about why we're not translating all of the things that we see in mice into humans yet, and a better understanding of some of these pathways that are relevant to NAD metabolism might really help us get there, considering things like intravenous dosing, or whether we really need to be using higher doses or local doses for certain indications, and really what the downstream pathways are that are relevant, which will be really important if we're really going to achieve some of the goals we have in humans for these NAD precursors. So clearly there's a lot left to learn about the fundamentals of NAD metabolism. And so with that, I'm done. I was asked to comment a little bit on my own habits with regard to supplements and NAD precursors. And I can say I don't actually have a regimen that I have adopted or committed to. I certainly experiment with NAD precursors once in a while just to see if I can feel any of the things I've heard anecdotally from people. I can tell you, for the most part, they've only made me not sleep as well. I feel like I have too much energy at bedtime sometimes if I've been taking them. But again, I don't really do anything consistently at this point, other than I also do tend to try to eat in a 12-hour window, which is more just my habit. I feel better when I do that. I really think, to be honest, that we have a lot more research to do with respect to NAD precursors to really be able to firmly commit to a science-backed regimen. And I hope that we're going to be able to get there. So with that, I just want to thank some of the people in my lab, and particularly point out Karthi Chalapa, who did a lot of the tracer studies that I showed you in collaboration with Melanie McReynolds. Tim Luongo did a lot of the mouse work and the mitochondrial transporter studies. I showed some data from Shamistha Mukherjee on liver regeneration. And of course, everyone here made major contributions to the work coming out of the lab over the last few years. And so thank you for listening. I hope you enjoyed the talk.